everyone, welcome to my channel Cells and Genes. So today we are going to discuss on the topic polymerase chain reaction. So what is polymerase chain reaction? We might have heard the term PCR a lot, so especially during the COVID times. So polymerase chain reaction, it is a molecular biology technique for selective amplification of DNA in vitro, it means inside in a closed system. So this is a revolutionary method which has been developed by Carey Mullis. Okay, it has been developed by Carey Mullis in 1980s, specifically saying in 1983. So it is mainly based on the ability, or it's based on using the ability of DNA polymerase enzyme to synthesize new strands of DNA that is complementary to the offered template strand. So if you provide them with one strand, DNA strands, it will synthesize multiple copies of the similar strands or multiple copies of the target strands. So let me give the definition. What is PCR? So PCR, or polymerase chain reaction, is a molecular biology technique for the in vitro amplification of specific DNA fragments, that is the targets, the specific DNA fragments. Since the advent of PCR technology, PCR has played a very important role in biological sciences, molecular diagnostics, we can say paternity testing, forensic identification, and crime investigation, and is one of the most important technologies that has been used in molecular biology, or that has been used in clinical diagnostics today. We have main three generations. We have three different types of PCRs. That is the normal conventional PCR, the next is quantitative PCR, and the next one is digital PCR. So mainly three generations are there. Now let's discuss the principles of PCR. So the basic principle is similar to the DNA replication in vivo. It mainly depends on the oligonucleotide primers. So it mainly depends on the primers that are complementary to both ends of the target sequence. So PCR mainly consists of three steps. One is denaturation, next is annealing, and extension. Denaturation of the DNA, or the template DNA, occurs around 95 degrees Celsius, where the double-stranded DNA is being heated to high temperature. It will be heated for a certain period of time, and we'll be fixing that temperature during the run, and it will break, or it will dissociate the double-stranded DNA into single-stranded, so that it will help the primers to bind. The next step is annealing. So annealing, the temperature is being lowered to 55 degrees Celsius. So in that step, we have the double-stranded DNA separated or denatured into two single strands. So in that step, in the annealing step, the primers bind to the single-stranded DNA, which is based on the principle complementary base pairing. The next step is primary extension, which is about 72 degrees Celsius. So this temperature is very important, because this is the temperature, or this is the optimum temperature for the activity of DNA polymerases. So when the temperature is set about 72 degrees Celsius, the DNA template and the primer binding are synthesized in the direction of phosphate to pentose in the 5' to 3' direction, and a new semi-conserved replication strand complementary to the template DNA strand under the action of Taq DNA polymerase, in accordance with the principle of base pairing and semi-conserved replication. So we get newly synthesized DNA. This amplifies the target gene to be amplified several million times after several cycles of denaturation, annealing, and extension repeats. So mainly we have three main steps. One is denaturation, the other one is annealing, the other one is primer extension. So let me show the diagrams. So this is the double-stranded DNA or the template DNA with the target sequence. So once the PCR starts, we have the first step, denaturation. So we have to separate the double-stranded DNA into single strands. So this occurs around 94 to 95 degrees Celsius. So once we get the single-stranded DNA, the next step is annealing. In the annealing procedure, we bring the temperature down, and then the primers add to this single-stranded DNA, and they bind by complementary base pairing. The next step is extension or synthesizing of new DNA strands at 72 degrees Celsius, where the DNA polymerase acts and the newly synthesized new strands are being synthesized. So this cycle is being repeated around 25 to 30 times, and we get copies or millions of copies of DNA with the target sequence. So let us discuss a few applications of PCR. The first one is prenatal diagnosis. Okay, the first one is prenatal diagnosis. Second one is detection of pathogens, which we have mainly applied in during the COVID times. The next one is genotyping. The next one, drug and discovery. And the next one is mutation screening. So these are the few applications of PCR, and like prenatal diagnosis, detection of pathogens, genotyping, and so on. There are plenty of applications, but these are few of them, which is easy to remember. So that was a quick video on what is PCR and what are the components and what are the different steps, and a short procedure, like applications. I have just mentioned like very few applications of PCR. So in the coming videos I will be uploading the different types of PCR and what are their applications, everything in detail. So thank you for watching. Please subscribe to Cells and Genes and look for more videos. Thank you.